Tonight's lecture is over Chapter 9, Genes and Inheritance. And our learning objectives are to be able to distinguish an organism's genotype from its phenotype. We will also be able to discuss how alleles segregate in meiosis. This is Mendel's law of segregation. We will learn how to use Punnett squares to predict the offspring of parents with particular genotypes. We will also be able to explain how a test cross can be used to determine the genotype of an organism. We will also be able to describe codominance, dominance, multiple alleles, polygenic inheritance, and pleiotropy. We will be able to describe how sex-linked traits are inherited, and we will be able to explain why some genes are linked together. Can a gene be cruel? Of course not. But if one could, consider this candidate. In humans, there is a gene for an enzyme called FMO3, flavin-containing monooxygenase 3, which breaks down a chemical in our bodies that smells like rotting fish. Some unfortunate individuals inherit a defective FMO3 gene and can't break down the noxious chemical. Instead, their urine, sweat, and breath excrete it, causing them to smell like rotting fish. Worst of all, because the odor comes from within their bodies, they cannot wash it away. Called fish odor syndrome, this disorder often causes those afflicted by it to suffer ridicule, social isolation, and depression. For individuals born with this, beyond their own suffering, there looms a scary question. Will I pass this condition on to my children? They might also wonder how they came to have the disorder, particularly if neither of their parents suffered from it. To address this concern, recall from Chapter 8 that sexually reproducing organisms carry 2 copies of every chromosome in every cell, except for the sex cells. For example, humans have 23 pairs of chromosomes, 46 individual chromosomes. At each location, referred to as a locus, or plural loci, on the 2 chromosomes of a pair is the same gene, 1 copy from the mother and 1 copy from the father. As we noted in a previous chapter, I believe chapter 6, each of the 2 copies of the gene is called an allele. Moving on, the gene for FMO3 is found on chromosome 1. Most people carry a functioning version or allele of the gene for FMO3, but there is also a rare, defective version that is responsible for fish odor syndrome. As long as a person has at least one functioning version of the FMO3 gene, he or she will produce enough of the enzyme to break down the fishy-smelling chemical. But if a person inherits 2 copies of the defective version of the fish odor gene, one from each parent, that person will inherit the disorder. Although individuals with fish odor syndrome will pass on one set of the bad instructions in every sperm or egg cell that they make, their children won't necessarily inherit the disease. As long as the other parent supplies a functioning version of the FMO3 gene, the child will not have fish odor syndrome. The unaffected children, though, will carry a silent copy of the fish odor allele. If they have children, the fish odor trait will be expressed only if it comes together with a defective FMO3 allele from another person. In this way, some alleles can exist in a population without always revealing themselves. In this chapter, we explore how heredity works. Inheritance follows some simple rules that allow us to make sense of patterns of family resemblance, such as facial features or hair texture, and even to predict the likelihood that an offspring will inherit a particular trait. We'll also examine why the behavior of some traits is easy to predict, while many other traits have less straightforward patterns of inheritance. And that brings us to our take-home message for section 9.1. Offspring resemble their parents because they inherit genes, instructions for biochemical, physical, and behavioral traits, some of which are responsible for diseases from their parents. So, like mother, like daughter, No one will ever seek a genetic test to prove that Zoe Kravitz is the daughter of Lisa Bonet. We see all around us that offspring generally resemble their parents more than they resemble other random individuals in the population. This is a consequence of the passing of characteristics from parents to offspring through their genes. And this concept is called heredity. Moving on, observing heredity is easy. Figuring out how it works is not. For thousands of years before the mechanisms of heredity were discovered and understood, plant and animal breeders recognized that there is a connection from parents to offspring across generations. In ancient Greece, for example, the poet Homer extolled the tremendous benefits to society that came from the skillful breeding of horses. Once breeders recognized the existence of heredity, they began selecting individual plants or animals with the desired traits to breed with each other in the hope that the offspring would also have those desirable traits. Since then, it has been possible to create a rich world of sweet corn, loyal dogs, docile livestock, beautiful flowers, and more. Heredity. I am the family face. Flesh perishes, I live on, projecting trait and trace through time and times anon, and leaping from place to place over oblivion. Thomas Hardy, in Moments of Vision and Miscellaneous Verses, 1917. Practical successes were many, but people didn't understand exactly how the outcomes were achieved. Patterns of similarity among related individuals are impossible to make sense of without an understanding of how heredity works. This understanding is the province of the field of genetics. Moving on, the mechanisms of heredity are easier to grasp when we focus on visible traits with well-established inheritance patterns, such as coat color and fur length in cats. Virtually every short-haired cat, for instance, has at least one short-haired parent, and virtually every completely white-haired cat has at least one white-haired parent. These are important observations that suggest likely features of the trait's mode of inheritance. In human, many genetic diseases have similarly simple patterns of inheritance. Individuals who develop Huntington's disease, for example, have a parent who developed the disease. Traits that are determined by the instructions a person carries on one gene are called single gene traits. However, most human characteristics are actually influenced by multiple genes as well as by the environment. And because the mechanism by which single gene traits pass from parent to offspring is the easiest pattern of inheritance to decipher, we will first explore this process. And that brings us to our take-home message for section 9.2. Some traits are determined by instructions that an individual carries on a single gene, and these traits exhibit straightforward patterns of inheritance. So, what do parents give their offspring that confer similarity? Well, in the mid-1800s, Gregor Mendel, a monk living in what is now the Czech Republic, was the first to make headway in answering this question. Indeed, Mendel conducted studies that almost completely answered the question. When Mendel turned to questions about heredity, there were no obvious answers. A popular idea from the late 17th century suggested that an entire pre-made human, albeit a tiny one, was contained in every sperm cell. Though imaginative, this idea failed to explain why children resemble both their mothers and their fathers rather than just their fathers. Also, a blended blood idea suggested that traits are blended in the blood of parents and imparted to offspring. But how, then, could brown-eyed parents give birth to blue-eyed children? Well, three aspects of his research were crucial to Mendel's success. First, he wisely chose to study the garden pea. Mendel's goals were to understand inheritance in all organisms, not just plants. Cats and dogs or even mice would have been too hard to take care of in the large numbers he required, thousands and thousands of individuals. By comparison, Pea plants are relatively easy to fertilize manually by quote-unquote pollen dusting. A single cross, the process in which male pollen carrying sperm is used to fertilize eggs, produces numerous offspring. In addition, pea plants reproduce quickly, so Mendel could conduct experiments that included multiple generations. Secondly, Mendel focused on easily categorized traits like shape and color. All peas of the variety that Mendel studied are either round or wrinkled, with nothing in between. In addition, all peas are either yellow or green in color, without any intermediate shade. In all, Mendel looked at 7 different traits of the pea plants, but for each trait, only 2 easily identified variants ever appeared. Thirdly, Mendel began his studies by repeatedly breeding together similar plants until he had many distinct populations, each of which was unvarying for a particular trait. He described these plants as true breeding for the trait because they always produced offspring with the same variant of the trait as the parents. For example, when true breeding round pea plants were crossed together, they always produced plants with round peas. True breeding purple flowered pea plants always produced purple flowered offspring. Mendel began a straightforward and methodical process of experimentation with his groups of true breeding plants. He crossed plants with different traits and observed large numbers of their offspring. For example, he crossed plants with green peas and plants with yellow peas and counted the number of offspring that produced green peas and the number that produced yellow peas. This cross, written as green times yellow cross, always resulted in plants that produced only yellow peas. Mendel devised a hypothesis that would explain his observations and generate predictions about the outcome of further crosses. Then he conducted those crosses to see whether the predictions generated by his hypothesis were borne out. There was elegance to Mendel's work. From his predictions, he articulated simple questions that could be answered by his experiments. For example, if I cross a plant that produces wrinkled peas with a plant that produces smooth peas, will the offspring plants produce wrinkled or smooth peas? His crosses would come out either one way or the other. This process of performing well-thought-out, rigorous experiments was a radical innovation for biology in Mendel's time. And that brings us to our take-home message for section 9.3. In the mid-19th century, Gregor Mendel conducted studies that help us understand heredity. He focused on easily observed and categorized traits in garden peas and applied methodical experimentation and rigorous hypothesis testing to determine how the traits are inherited. So Mendel was motivated by one odd and recurring result. Sometimes traits that weren't present in either parent pea plant would show up in their offspring, just as brown-eyed parents can have blue-eyed children. When plants with purple flowers, those not in his true breeding group, were fertilized by pollen from other plants with purple flowers, they produced mostly purple-flowered offspring, but sometimes they produced plants with white flowers. How was it possible to produce white flowers from a purple-purple cross? Where did the whiteness come from? Here's why Mendel's meticulous and methodical experiments paid off. First, he started with some true-breeding white-flowered plants. Then he got some true-breeding purple-flowered plants. He wondered which color wins out when a white-flowered plant is crossed with a purple-flowered plant. The answer was definitive. The purple flower wins. All of the offspring from these crosses were purple every time. For this reason, Mendel called the purple flower trait dominant, and he considered the white flower trait to be the recessive trait. In general, a dominant trait masks the effect of a recessive trait when an individual carries both the dominant and the recessive versions of the instruction for the trait. But things got a little bit more interesting when Mendel took the purple-flowered plants that came from the cross between the true-breeding purple-flowered and white-flowered plants and bred these purple offspring with each other. He found that these mixed parentage plants were no longer true-breeding. Occasionally, they would produce white-flowered offspring. So apparently, the directions for building white flowers, last seen in one of their grandparents, were still lurking inside the purple-flowered parent plants. This existence of traits that could disappear for a generation and then show up again was perplexing. Moving on, Mendel devised a simple hypothesis to explain this pattern of inheritance. It incorporated 3 ideas that helped him and now help us make predictions about crosses. 1. Rather than passing on the trait itself, each parent puts into every sperm or egg it makes a single set of instructions for building the trait. Today we call that instruction set a gene. 2. Offspring receive 2 copies of the instructions for any trait. Often, both sets of instructions are identical, for example, a purple-purple cross, and the offspring produce the trait according to those instructions. At other times, each parent contributes a slightly different set of instructions, that is, a different allele for that trait. So, for example, pea plants have 2 alleles for flower color, a purple flower allele and a white flower allele. Parents pass on the same gene or flower color gene, but may contribute different alleles, which could be purple or white. Thirdly, the trait observed in an individual depends on the 2 copies of gene that it inherits from its parents. When an individual inherits the same 2 alleles for this gene, the individual's genotype for that gene is said to be homozygous, and the individual shows the trait specified by the instructions embodied in those alleles. When an individual inherits a different allele from each parent, the individual's genotype for that gene is said to be heterozygous. Dominant and recessive alleles are defined by their action when they are in the heterozygous state. Specifically, a dominant allele masks the effect of the other allele, which is called the recessive allele. Note that this is the sole defining feature of dominant and recessive alleles. So describing an allele as dominant does not mean it is more advantageous or more common than a recessive allele. Moving on. When an individual reproduces, it contributes just one of its 2 copies of a gene to its offspring. The other parent contributes the other allele. So, when sperm and egg are made, each sex cell gets only 1 copy of a gene as opposed to 2 copies present in every other cell in the body or present in the somatic cells. So for a male who is heterozygous for a particular gene, half of the sperm he produces will have 1 of the alleles and half will have the other allele. The idea that of the 2 copies of each gene everyone carries, only 1 of the 2 alleles gets put into each gamete is so important that it is called Mendel's law of segregation. And that brings us to our take-home message for section 9.4. Each parent passes along in every sperm or egg that he or she makes an instruction set for building a particular trait. This instruction set is called a gene. The trait observed in an individual depends on the 2 copies of alleles of the gene inherited. So, things are not always as they appear. Take skin coloration, for example. Humans and many other animals have a gene that contains the information for producing melanin, one of the chemicals responsible for giving skin its coloring. This gene is one of many that influence skin color. Sometimes a defective, non-functioning version of the melanin gene is passed along through families. An individual who inherits 2 copies of the defective version of the gene cannot produce melanin and has a condition known as albinism. This is a disorder characterized by little or no pigmentation in the eyes, hair, and skin. A typically
pigmented individual may carry one of these defective alleles. However, we would need to do a genetic analysis to discover this information. The outward appearance and behavior of an individual is called its phenotype. A phenotype includes features visible to the naked eye, such as body size or skin pigmentation, or bright colored feathers, or the presence of antlers. A phenotype also includes less easily visible characteristics, such as the chemicals an individual produces to clot blood or digest lactose. Underlying the phenotype is the genotype, an organism's genetic composition. We usually speak of an individual's genotype in reference to a particular trait. For example, an individual's genotype might be described as homozygous for the recessive allele for albinism, meaning that the person has two recessive alleles of the gene. Another individual's genotype for the melanin gene might be described as heterozygous, a recessive allele and a dominant allele. Occasionally, the word genotype is also used as a way of referring to all of the genes an individual carries. So I like to think of it like this. The phenotype is the characteristic that penetrates. Right? It's the characteristic that you can see, or the characteristic that you can observe. Because remember, like blood type is a phenotype, but you can't really look at a person and tell what blood type they are. Moving on. When an organism exhibits a recessive trait, such as albinism, we know with certainty what its genotype is for the melanin gene. When it shows the dominant trait, on the other hand, it's not possible to discern the genotype from the individual's appearance. In such a case, a handy tool called a Punnett square can help us identify the possible outcomes for crosses between two individuals. To see how a Punnett square works, let's investigate the cross of an albino giraffe with a normally pigmented giraffe. First, we assign symbols to represent the different variants of a gene. It is customary to use an uppercase letter for the dominant allele and a lowercase letter for the recessive allele. In the case of pigmentation, albinism, we use the letter M for production of the pigment melanin. Capital M for the dominant allele and lowercase m for the recessive allele. We represent the genotype of the albino giraffe as lowercase m, because that individual must carry two copies of the recessive allele. The pigmented giraffe must have the genotype capital N, capital M, or capital M, lowercase m. If we don't know which of the two possible genotypes the pigmented individual has, we can write uppercase M underscore, where underscore is a placeholder for the unknown second allele. In this figure on your slide, 9.11 on page 247, it illustrates the cross between true breeding pigmented individuals with capital M, or homozygous dominant, and an albino individual, lowercase m, or we could say homozygous recessive. Along the top of the Punnett square, we list individually the two alleles that one of the parents produces, and along the left side of the square, we list the two alleles that the other parent produces. We split up an individual's alleles in this way because only one of the alleles is contained in each sperm or egg cell that it produces. The two gametes that come together at fertilization produce the genotype of an offspring. In the four cells of the Punnett square, we enter the genotypes of all possible offspring resulting from our cross. Each cell contains one allele given at the head of the column and one allele at the left row of the column. In cross 1, shown in this figure, every possible offspring would be heterozygous and normally pigmented, because all offspring would receive a dominant allele from the pigmented parent and a recessive allele from the albino parent. In the bottom half of this figure, cross 2, we trace the cross between two heterozygous individuals. Note that each parent produces two kinds of gametes, one with the dominant allele and one with the recessive allele. This cross has three possible outcomes. 1/4 of the time, the offspring will be homozygous dominant, or capital M, capital M. 1/4 of the time, the offspring will be homozygous recessive. Remember, that's lowercase m, lowercase m. And the remaining half of the time, 50% of the time, the offspring will be heterozygous. In other words, capital M, lowercase m. Phenotypically speaking, 3/4 of the offspring will be normally pigmented. They will either be homozygous recessive or heterozygous for this allele, and 1/4 will be homozygous recessive. And that brings us to our take-home message for section 9.5. It is not always possible to determine an individual's genetic makeup, known as its genotype, by observing the organism's outward appearance, known as its phenotype. For a particular trait, an individual may carry a recessive allele whose phenotypic effect is masked by the presence of a dominant allele. So, even with perfect information, it can still be impossible to know a genetic outcome with certainty. It's like flipping a coin. You can know every last detail about the coin, but you still can't know whether a coin will land on heads or tails. The best you can do is define the probability of each possible outcome. The rules of probability have a central role in genetics for two reasons. First, Mendel described the process of segregation, in which each gamete receives only one copy of each gene present in somatic cells. As a result, for each gene the individual carries, the likelihood that the sperm or egg will include one allele is the same as the likelihood that it will include the other allele. It is impossible to know which allele it will be. Second, as we pointed out in Chapter 8, all of the sperm or eggs produced by an individual are genetically different from one another, and any one of those gametes may be the gamete involved in fertilization. Therefore, like segregation, fertilization is a chance event. Knowing everything about the alleles that a parent carries is not always enough to be able to determine with certainty which alleles his or her offspring will carry. In games of chance, as well as in matings, we can make predictions based on probabilities. If an individual is homozygous for a trait, 100% of his or her gametes will carry that allele. However, each of the gametes of an individual for heterozygous traits has a 50% probability of carrying the dominant allele and a 50% probability of carrying the recessive allele. So let's use these probabilities in an example. What is the probability that an offspring will be homozygous for albinism, that is, lowercase m, if the male parent is heterozygous for albinism and the female parent is albino? To get to the MM outcome, two events must occur. First, the father's gamete must carry the recessive allele, the lowercase m. And second, the mother's gamete must carry the recessive allele, again, the lowercase m. In this case, the probability of a homozygous recessive offspring is 0.5. The probability that the father's gamete carries the lowercase m, or the recessive trait, times 1, which is the probability that the mother's gamete carries the recessive trait. This yields a probability of 0.5, 50%, 1/2, or 1 in 2, however you want to term it. This is a general rule when determining the likelihood of a complex event occurring. If you know the probability of each component that must occur, you multiply the probabilities together to get the overall probability of that complex event occurring. Moving on, consider an example that involves Tay-Sachs disease. As described in a previous chapter, Tay-Sachs disease is caused by malfunctioning lysosomes that do not digest cellular waste properly and leads to death in early childhood. Tay-Sachs disease occurs when a child inherits two recessive alleles for the Tay-Sachs gene. If each parent is heterozygous for the Tay-Sachs gene, what is the probability that their child will have the disease? Well, let's think about it. Half of the father's sperm will have the dominant allele, and half will have the recessive allele. So the probability of the child inheriting the recessive allele is 0.5. And half of the mother's eggs will have the dominant allele, while the other half will have the recessive allele. So the probability of the child inheriting the recessive allele from its mother is also 0.5. Thus, the overall probability of the child having Tay-Sachs disease is 0.5 times 0.5, which equals 0.25, or 1 in 4, 1/4. Of course, if the couple has only one child, we can't predict with certainty whether the child will have Tay-Sachs disease. The mathematical probability tells us, however, that if the couple had an infinite number of children, we could expect that 1/4 of them would have the disease. This brings us to our take-home message for section 9.6. Probability plays a central role in genetics. In segregation, each gamete receives only one of the two copies of each gene the individual carries in its somatic cells. But it is impossible to know which allele goes into the gamete. Chance plays a role in fertilization too. All gametes produced by an individual are different from one another, and any one may be the gamete involved in fertilization. So, suppose you are in charge of the alligators at a zoo, and some of your alligators come from a population in which white albino alligators have occasionally occurred. Although none of your alligators is white, because white alligators are popular with zoo visitors, you would like to produce some at your zoo through a mating program. How would you go about producing white alligators? Well, the problem is that you cannot be certain of the genotype of your alligators. They might be homozygous dominant, or they might be heterozygous. In either case, their phenotype is typical coloration. Determining the genotype of a particular alligator is a challenge to animal breeders, but not an insurmountable one. Genes may be visible, but their identity can be revealed by a tool called the test cross. Moving on. In the test cross, you cross or mate an individual exhibiting a dominant trait but whose genotype is unknown with an individual that is homozygous recessive. Then you examine the phenotypes of their offspring. To breed an albino alligator, you could borrow an albino alligator from another zoo and breed your unknown genotype alligator with an albino alligator. There are two possible outcomes, and they will reveal the genotype of your M_alligator. If your alligator is homozygous dominant, it will contribute a dominant allele to every offspring. Even though the albino alligator will contribute the recessive allele to all of its offspring, the offspring of this cross will be heterozygous, and none of them will be albino. If, on the other hand, your M_alligator is heterozygous, half of the time it will contribute a recessive allele to the offspring. In every one of those cases, the offspring will be homozygous recessive and thus albino. To be confident that the unknown genotype alligator has the dominant genotype, though, you'd have to observe many, many offspring. After all, even if its genotype is heterozygous, quite a few offspring in a row might be typically pigmented with the genotype for heterozygosity, which is capital M lowercase m. Eventually, however, a heterozygous individual is likely to produce an offspring with the homozygous recessive genotype and the albino phenotype. For this reason, a test cross can identify an individual's genotype much more quickly in fruit flies, which can lay 100 or more eggs per day, than in species with slower reproductive rates. And that brings us to our take-home message for section 9.7. In a test cross, an individual that exhibits a dominant trait but has an unknown genotype is mated with an individual that is homozygous recessive. The phenotypes of the offspring reveal whether the unknown genotype individual is homozygous dominant. All of the offspring exhibit the dominant trait, or heterozygous. Half of the offspring show the dominant trait, and half show the recessive trait. So, prospective parents may want to know the likelihood of having a child with a particular genetic disease, say hemophilia. Geneticists who study diseases may want to know how a particular disease or trait is inherited. Is it recessive or dominant? Is it carried on the sex chromosomes or on the autosomes? A pedigree is a type of family tree that can help answer these questions. For instance, dog and horse breeders often speak of pedigreed animals, a feature that adds tremendous value to the animals. In particular, with knowledge of an animal's family tree, it is much less likely that any genetic surprises will occur as the animal develops and reproduces. In a pedigree, information is gathered from as many related individuals as possible across multiple generations. Starting from the bottom, each row in the chart represents a generation listing all of the children in their order of birth, their sex, and whether they have a particular trait. Working up the pedigree, the children's parents are indicated, and above them, their parents' parents, for as far back as the data is available. Squares represent males and circles represent females, and these shapes are shaded to indicate that an individual exhibits a trait of interest. Sometimes a genotype, or as much of it as is known, is also listed for each individual. The pedigree is analyzed for patterns of inheritance. For dominant traits, all affected individuals must have at least one parent who exhibits the trait. In contrast, for recessive traits, an individual may exhibit a recessive trait even if neither parent shows the trait. In this case, the individual's parents must be heterozygous for that trait. The pedigree can also help determine whether a trait is carried on one of the sex chromosomes, the X or the Y chromosome, or on one of the non-sex chromosomes that we call the autosomes. Traits that are controlled by genes on the sex chromosomes are called sex-linked traits. Recessive sex-linked traits, for example, appear more frequently in males than in females, whereas dominant sex-linked traits appear more frequently in females. These patterns may become obvious only on inspection of a large pedigree. Anuria is a condition seen in dogs and some other animals in which the animal has no tail. The pedigree reveals that anuria is inherited as a recessive trait because unaffected parents can have offspring without the disorder. Can you determine the genotype of the individual labeled number 1 in figure 9.16 on page 252? Why must that female be heterozygous for anuria? By the way, an individual that carries one allele for the recessive trait and so does not exhibit the trait but can have offspring that do is referred to as a carrier of the trait. Must her mate have the same genotype? On the same pedigree, note that 2 individuals in the second row of the pedigree have a puppy indicated by a question mark. What is the probability that this puppy has anuria? Examine the pedigree carefully and see whether you can come up with the answer. Well, in the cross producing the mystery puppy, the father has anuria and so must be homozygous recessive. He will definitely pass on one recessive allele to the son. On the other hand, the mother's genotype can be homozygous dominant or heterozygous, given that both of her parents are heterozygous. Consequently, she has a 2/3 probability of being a carrier of the recessive allele. If she is, then there is a 50% chance that she will pass the allele on to her offspring and he will have anuria. The probability, therefore, is 2/3 times 1/2, or 1/3, or a 1 in 3 chance that the puppy will have a nuri. As we'll discuss later in this chapter, some traits may not show complete dominance, and many traits are also influenced by the environment, so a trait's mode of inheritance is not always obvious. In such cases, the more individuals we can include in the pedigree, the more accurate the analysis will be. Some human pedigrees contain thousands of individuals and stretch back 6 or more generations. With some other species, it is possible to analyze tens of thousands of individuals per generation for a dozen or more generations. This is why plants and small insects, among other organisms, are often a better choice for studying inheritance patterns. Once we have an idea about how a trait is inherited, we can identify individuals who are carriers of a recessive trait and make informed predictions about a couple's risk of having a child with a particular disorder. This knowledge can be useful in conjunction with prenatal testing and treatment. And that brings us to our take-home message for Section 9.8. Pedigrees help scientists, doctors, animal and plant breeders, and prospective parents determine the genes that individuals carry and the likelihood that the offspring of the two individuals will exhibit a given trait. So, as Mendel saw it, the world of genetics was straightforward. Each of the traits he studied was coded for by a single gene with 2 alleles, one that is completely dominant and one that is recessive and with no environmental effects. We should be so lucky. In this and the following sections, we build up a more complex model of how genes influence the building of bodies. First, we begin with the observation that the phenotype of heterozygous individuals sometimes differs from that of either the homozygous for the trait and instead reflects the influence of both alleles rather than a clearly dominant allele. Let's look at the case of incomplete dominance in which the phenotype of a heterozygote is intermediate between the phenotypes of the two homozygotes. Moving on, an example of incomplete dominance that we can easily observe is the flower color of snapdragons. We can obtain true breeding homozygous lines of snapdragons with red flowers and true breeding homozygous lines that produce only white flowers. Given what we learn from Mendel's experiments, when plants from these two populations are crossed, we expect a generation with either all red or all white flowers. However, such crosses with snapdragons always produce plants with pink flowers. And when we cross 2 plants with pink flowers, we get 1/4 red-flowered plants, 1/2 pink-flowered plants, and 1/4 white-flowered plants. So how can we interpret this cross? Here, we use a slightly different way of denoting genotype. We use the letter C to indicate a gene that codes for color, and the superscript W or R refers to an allele producing no pigment, white, or red pigment. Thus, the plants with white flowers have the genotype C superscript W, C superscript W, and produce no pigment. At the other extreme, the plants with red flowers have the genotype C superscript R, C superscript R, and produce a great deal of pigment. We use these designations for the genotypes because it isn't clear that either white or red is dominant over the other, and so neither should it be represented as such. The plants with pink flowers receive one of the pigment-producing C superscript R alleles and one of the no pigment-producing C superscript W alleles, and so produce an intermediate amount of pigment. Ultimately, the intensity of pigmentation just depends on the amount of pigment chemical that is made by the flower color genes. An example of incomplete dominance in humans can be seen in the processing of cholesterol in the bloodstream. There is a membrane receptor that allows cells, chiefly those of the liver, to remove cholesterol from the bloodstream. Individuals who carry 2 copies of a mutant allele, called FH for this gene produce almost no low-density lipoprotein or LDL receptors. Consequently, in these individuals, circulating cholesterol levels are high and cardiovascular disease develops at a very young age. Few individuals survive past 20 years of age. This is in sharp contrast to individuals carrying 2 copies of the allele for normal functioning LDL receptors who experience significantly lower levels of circulating cholesterol. Heterozygotes carrying 1 copy of the mutant FH allele and 1 copy of the allele for normal functioning LDL receptors represent an intermediate situation. They produce about half as many LDL receptors as do people who are homozygous for the normal functioning receptor, leading to higher than average levels of circulating cholesterol and 10 times the risk of death from cardiovascular disease. Moving on, a second situation in which complete dominance is not observed is called codominance, in which the heterozygote displays characteristics of both homozygotes, as playfully represented in this figure on your slide. This is figure 9.18 on page 254, although the shirt phenotype, of course, has no genetic basis. In codominance, neither allele masks the effect of the other. An example of codominance occurs with feather color in chickens. When white chickens are crossed with black chickens, all of the offspring have both white and black feathers. And that brings us to our take-home message for section 9.9. With incomplete dominance, the phenotype of a heterozygote appears to be an intermediate blend of the phenotypes of the 2 homozygotes. With codominance, a heterozygote has a phenotype that exhibits characteristics of both homozygotes. So, do you know your blood type? It can be O, A, B, or AB. Each of these blood types, also called blood groups, indicates something about the physical characteristics of your red blood cells and has implications for blood transfusions, both giving and receiving blood. The blood groups are interesting from a genetic perspective because they illustrate the case of multiple allelism, in which a single gene has within a population more than 2 alleles. Of course, each individual still carries only 2 alleles, one from the mother and one from the father. Inheritance of the ABO blood groups provides the simplest explanation of multiple allelism because there are only 3 alleles. We can call these alleles I superscript A, I superscript B, and lowercase I, using both types of allele notation introduced earlier. The I superscript A and I superscript B alleles are both completely dominant to lowercase I. So individuals are considered to have blood type A, whether they have genotype I superscript A, I superscript A, or I superscript A lowercase I. And this can be seen on the figure on page 255, figure 9.19. Similarly, an individual with the genotype I superscript B, I superscript B, or I superscript B lowercase I is considered to have blood type B. If you carry 2 copies of the lowercase allele I, you have the blood type O. The I superscript A and I superscript B alleles are codominant with each other, so the genotype I superscript A, I superscript B gives rise to blood type AB. Consequently, with these 3 alleles in the population, individuals can be one of 4 different blood types, A, B, AB, or O. Moving on. These blood type alleles direct the production of a specific set of chemicals called antigens. Antigens are molecules, chiefly carbohydrates bound to protein, that jut out from the surface of a cell and can quote-unquote turn on a body's defense against foreign invaders. We label this gene with the letter I, capital I, to indicate isoagglutination, which is another word for antigen. The I superscript A allele directs the production of A antigens that cover the surface of red blood cells. Similarly, the I superscript B allele directs the production of B antigens on all red blood cells. Individuals with type AB have red blood cells with both A and B antigens. The lowercase I allele, or the recessive allele, does not code for the A antigen or the B antigen. This means that individuals with blood type O have red blood cells that have neither A nor B antigens on their surface. Antigens are like signposts in the body's disease-fighting immune system, telling the immune system whether a cell belongs in the body. If a red blood cell with the wrong antigens enters your bloodstream, your immune system recognizes it as a foreign invader and destroys it. Such an attack is initiated by molecules in the bloodstream called antibodies. Individuals with only A antigens on their red blood cells produce antibodies that attack B antigens. Under normal circumstances, antibodies do not encounter a red blood cell with foreign antigens. But such an event could occur if red blood cells with foreign antigens were accidentally injected into the person's bloodstream in a transfusion. An improper transfusion can cause destruction of red blood cells, low blood pressure, and even death. Individuals with only B antigens on their red blood cells produce antibodies that attack A antigens. Individuals with blood type O who have neither A nor B antigens on their red blood cells produce antibodies that attack both A and B antigens. And individuals with blood type AB don't produce either type of antibody, or else they would have antibodies that attack their own blood cells. Moving on, from this information, we can deduce which blood types can be used in transfusions. Individuals with blood type O are universal donors because their red blood cells have no A or B antigens and so do not trigger a reaction from either type of antibody. Conversely, individuals with blood type AB are universal recipients because they do not produce antibodies to either the A or B antigen. Moving on, another marker on the surface of red blood cells is the Rh blood group marker. Note that the Rh blood group is not an example of multiple allelism because a single gene with just two alleles determines the presence of the Rh marker. However, like the ABO blood groups, the Rh marker restricts the type of blood a person can receive in a transfusion. Individuals who possess red blood cells that carry the Rh cell surface marker have one or two copies of the dominant Rh marker allele and are said to be Rh positive. This positive is noted along with their ABO blood type, as in O positive, for example. Individuals who have two copies of the recessive allele for this gene do not have any Rh markers and are described as negative, as in O negative, for example. So if, during a blood transfusion, individuals who are Rh negative are exposed to Rh positive blood, their immune system attacks the Rh antigens as foreign invaders. An immune response that can vary from mild, which passes unnoticed, to severe, which can lead to death. So the Rh and Rh marker, sometimes described as Rh factor, was first discovered in the blood of rhesus monkeys. So that's where the Rh comes from, is from the word rhesus. According to statistics, 85% of all people living in the world are Rh positive, and 15% of people living in the world are Rh negative. And that brings us to our take-home message for section 9.10. In multiple allelism, a single gene has more than two alleles. Each individual still carries only two alleles, but in the population, more than two alleles exist. This is the case for the ABO blood groups in humans. So there's this old wives' tale that suggests a couple of ways that parents can predict the ultimate height of their child. If the baby is a boy, they say to add 5 inches to the mother's height and average that with the father's height. If it is a girl, subtract 5 inches from the father's height and average that with the mother's height. Alternatively, the lore says just take the child's height at 2 years of age and double it. These prediction methods can be surprisingly accurate because genes play a strong role in influencing height. Offspring do resemble their parents in measurable ways, but unlike Mendel's pea plants, in which a single gene with 2 alleles determines the height of a plant, for humans and most animals, adult height, a continuously varying trait, is influenced by many different genes. Such a trait is said to be polygenic. Recent research has identified at least 180 heritable loci that influence adult height. For example, of these loci, the alleles that a person carries play a role in determining height. Individuals with tall variants for more of the genes tend to be taller than those with tall variants for fewer genes. Many height genes play roles in skeletal growth and hormone pathways. For example, a gene on chromosome 15 codes for an enzyme that converts testosterone to estrogen. This enzyme influences height because estrogen helps bones fuse at their ends and thus stop growing. The term additive effects describes what happens when the effects of the alleles of multiple genes all contribute to the ultimate phenotype. The variety of heights among humans reflects the fact that height is a trait influenced by contributions from multiple genes as well as by the environment. Many other physical traits are influenced by multiple genes, including eye color in humans. Eye color was long considered a discrete trait, with just 2 distinct phenotypes possible, and believed to be controlled by a single gene with a dominant brown eye allele and a recessive blue eye allele. Now it seems that eye color is the result of interactions between at least 2 genes and possibly more, a situation that makes more sense given the continuous variations in eye color among human adults. Moving on, many behavioral traits are influenced by multiple genes. The developmental disorder known as autism, for example, seems to be the result of alterations in numerous, even as many as 100 genes. Individuals with autism have difficulty interacting with others, particularly in making emotional connections. They also tend to have narrowly focused and repetitive interests. Autism is notoriously difficult to study because its symptoms are so varied, as is their intensity. These variations may result from different combinations of the many genes involved in autism. The alleles responsible for autism may also influence some desirable characteristics. This idea arose when researchers documented an overrepresentation of autism among children of parents working in the fields of engineering, physics, computer science, and math. The researchers note, however, that children with autism are born to parents across all professions and socioeconomic levels. So that brings us to our take-home message for section 9.11. Many traits, including continuously varying traits such as height and eye color, are influenced by multiple genes. So, just as multiple genes can influence one trait, some individual genes can influence multiple unrelated traits, a phenomenon that we call pleiotropy. In fact, this may be true of nearly all genes. Consider sickle cell disease, also called sickle cell anemia, a potentially fatal condition in which individuals produce defective red blood cells that become sickle-shaped when they lose the oxygen they carry. The defective blood cells can't effectively transport oxygen to tissues, and they accumulate in blood vessels, causing extreme pain. Individuals with sickle cell disease suffer from shortness of breath and numerous other problems that lead to a significantly reduced lifespan. The gene responsible for sickle cell disease codes for part of the hemoglobin molecule, the oxygen-carrying molecule in red blood cells. Capital H, lowercase b, superscript a is the allele for normal hemoglobin, and capital H lowercase b superscript s is the abnormal sickle cell allele. Sickle cell anemia occurs in individuals homozygous for the sickle cell allele, which would be capital H lowercase b superscript s, capital H lowercase b superscript s, but not in individuals carrying at least one copy of the normal allele, which is capital H lowercase b superscript a. Heterozygous individuals produce both normal and sickling red blood cells, but not enough sickle-shaped cells to cause sickle cell anemia. This hemoglobin gene is pleiotropic because, although it is just one gene, it causes multiple phenotypic effects. Individuals homozygous or heterozygous for the sickle cell allele have abnormal hemoglobin and red blood cells as well as circulatory problems. Moreover, individuals with these genotypes are resistant to the parasite that causes malaria. The malaria parasite, which lives in red blood cells, cannot survive well in cells that carry the defective version of the hemoglobin gene. Another example of pleiotropy involves the gene called CFTR, which codes for a membrane protein that serves as a channel for chloride ions. Mutations that impair the functioning of these ion channels result in the disease cystic fibrosis with its characteristic buildup of mucus in the lungs. They also give rise to diabetes resulting from reduced pancreatic functioning and in males cause a deterioration of the vas deferens leading to infertility. And that brings us to our take-home message for section 9.12. In pleiotropy, one gene influences multiple unrelated traits. Most, if not all, genes may be pleiotropic. So, the patterns of inheritance of most traits do not differ between males and females. When a gene is on an autosome, or one of the non-sex chromosomes, both male and females inherit two copies of the gene, one from their mother and one from their father. The likelihood that an individual inherits one genotype rather than another does not differ between males and females. In contrast, traits coded for by the sex chromosomes have different patterns of expression in males and females. One of the most easily observed examples of this phenomenon is red-green color blindness. The X chromosome in humans has a gene that carries instructions for producing the light-sensitive proteins in the eye that enable us to distinguish between the colors red and green. If an individual has at least one functioning copy of this gene, he or she produces sufficient amounts of the protein to have normal color vision. A rare allele for this gene, however, produces a non-functioning version of the light-sensitive protein. Having some of this non-functioning protein is not a problem if the person also carries another normal version of the gene and produces some of the functioning protein. But here's the problem. Men get only one chance to inherit the normal version of the gene that codes for red-green color vision. Because the gene is found on the X chromosome, men inherit this chromosome only from their mother. Women get two chances. Although a woman may inherit the defective allele from one parent, she still can inherit the normal allele from the other parent and have normal color vision. If she inherits the defective allele from both parents, she will be red-green colorblind. As we would then predict, the frequency of red-green color blindness is significantly greater in males than in females. Approximately 7% to 10% of men exhibit red-green color blindness, whereas fewer than 1% of women have this condition. Although men exhibit sex-linked recessive traits more frequently than women do, the situation is reversed for sex-linked dominant traits. In these cases, because females have two chances to inherit the allele that causes the trait, they are more likely to have the allele and thus exhibit the trait than are males, who only have one chance to inherit the allele. I would also like to point out here that since women have two X chromosomes, their alleles can be either homozygous or heterozygous. But since men only have one X chromosome and one Y chromosome, men are what we refer to as hemizygous for those traits. And that brings us to our take-home message for section 9.13. When a trait is coded for by a gene on a sex chromosome, such as color vision on the X chromosome, the pattern of expression differs for males and females. So, it is a very serious warning emphasized in bold capital letters. Phenylketonurics contains phenylalanine. But it's not placed next to a skull and crossbones like you would see on a glass bottle in a chemistry lab. Instead, this warning is found on cans of diet soda. The warning is intended for people with a genotype that, if they consume the amino acid phenylalanine, can be deadly. Sometimes our bodies use phenylalanine directly to build proteins, adding it to a growing amino acid chain. At other times, phenylalanine is chemically converted into the amino acid tyrosine. The body may then use tyrosine as it constructs proteins and in a variety of other functions. The problem is this: at birth, some people carry 2 copies of a mutant version of the gene that is supposed to code for the enzyme that converts phenylalanine to tyrosine. The mutant gene produces a malfunctioning enzyme so that none of the body's phenylalanine is converted into tyrosine. Little by little, as these individuals consume phenylalanine, it builds up in their bodies. Babies born with 2 mutant alleles usually begin to show symptoms within 3 to 6 months. Within a few years, so much phenylalanine accumulates in their bodies that it reaches toxic levels and poisons the brain, leading to mental retardation and other serious health problems. This disease is called phenylketonuria, or PKU. Here's where the warning label comes in. By limiting the amount of phenylalanine consumed, individuals with 2 mutant alleles for processing phenylalanine can avoid the toxic buildup of the amino acid in their brains. In essence, they modify their environment or their diet so that it contains only a tiny, carefully monitored amount of phenylalanine. Because all newborn babies in the United States are screened for PKU at birth, they can be treated with the appropriate diet immediately. And in an environment free of excess phenylalanine, the PKU mutant alleles are harmless. This example highlights the fact that genes interact with the environment to produce physical characteristics. Unless you have information about both genes and the environment, it is not usually possible to know what the phenotype will be. Moving on, Siamese cats carry genes that produce dark pigmentation. These genes interact strongly with the environment and are heat sensitive. Cold areas of the animal's body produce dark pigment, while warm areas remain light in color. The coldest parts of the body, the ears, paws, tails, and tip of the face, become the darkest, while the rest remains cream-colored or white. Interestingly, Siamese cats that live in cold climates and spend a lot of time outside become significantly darker in color during the winter months, and those that lounge indoors all winter remain lighter in color. There are thousands of other cases in which genes' interactions with the environment influence their effects in the body. The scope of environmental influences ranges from traits with obvious environmental effects, such as body weight and its relationship with caloric intake, to those with slight environmental effects, such as fur color, to those with complex and subtle interactions with the environment, such as intelligence and personality. Given the role of environmental factors in influencing phenotypes, DNA is not like a blueprint for a house. There is nearly always significant interaction between the genotype and the environment that influences the exact phenotype produced. If this were not true, there would be no reason to invest in better schools, physical fitness regimens, nutritional monitoring, self-help efforts, or other processes by which individuals or societies try to improve people's lives, in other words, altering phenotypes by enriching their environment. And that brings us to our take-home message for section 9.15. Genotypes are not like blueprints that specify phenotypes. Phenotypes are a product of the genotype and its interactions with the environment. So sometimes you can be right about something for the wrong reason, and this happened to Gregor Mendel. Mendel didn't know that genes were carried on chromosomes. Instead, he believed that the units of heredity were just free-floating entities within cells. From this perspective, it made sense that the inheritance pattern of one trait wouldn't influence the inheritance of any other trait. The relation of each pair of different characters in hybrid union is independent of the other differences in the two original parental stocks. Gregor Mendel, clearly articulating the idea of independent assortment in his publication, Experiments in Plant Hybridization, 1865. But let's consider an example. Earlier in this chapter, we saw that all cats with completely white fur have at least one parent that also has completely white fur. This is because white fur is coded for by a single dominant gene. Imagine that you had a true breeding population of cats with white fur. In other words, all offspring manifest the trait, and in this case, have a homozygous dominant genotype. Now suppose that an individual in this population mated with a cat from a true breeding population of cats that all had some colored fur, all individuals with the recessive genotype. All of their offspring would have white fur, but they would be heterozygous. If two heterozygotes had offspring together, though, they would produce 3/4 white-furred and 1/4 non-white furred offspring with genotypes in the ratio of 1/4 homozygous dominant, 1/2 heterozygous, and 1/4 homozygous recessive. That is just what Mendel observed for traits in the pea plants. But what if we concurrently observed another characteristic of these cats? Suppose the original true breeding population of white-furred cats also true breeding for long-haired fur, a condition which is caused by two recessive alleles for a single gene. And suppose that individuals in the colored-furred population, in other words, the homozygous recessive population, were also true breeding for short hair. The question is, do the alleles an individual inherits for white fur trait influence which alleles that individual inherits for fur length? The answer is no, they do not. Rather, the first cross of a long-haired, all-white cat with a cat having short hair and colored fur would result in offspring heterozygous for both traits, referred to as a dihybrid, and would express each of the dominant traits. Moving on, phenotypically, all of the offspring from this cross would have short, completely white fur. In a mating between two of these doubly heterozygous individuals, referred to as a dihybrid cross, 3/4 of the offspring would have the dominant trait and 1/4 would have the recessive trait, regardless of which trait you are tallying. You can extract two single trait Punnett squares from the big 4x4 Punnett square. In other words, neither trait influences the inheritance pattern for the other trait, and all traits are inherited independently of each other. This is known as Mendel's law of independent assortment. And that brings us to our take-home message for section 9.16. Genes tend to behave independently such that, for most traits, the inheritance pattern of one trait does not influence the inheritance of any other trait. So most redheads have pale skin and freckles. This simple observation is problematic for the law of independent assortment as Mendel imagined it. But clearly, having red hair seems to influence the presence of another trait, pale skin. Strictly speaking, Mendel's second law is not true for every pair of traits. Sometimes the alleles for two genes are inherited together and expressed almost as a package. We have about 21,000 genes in our genome, yet we only have 23 unique chromosomes and 2 copies of each. Thus, genes influencing different traits must sometimes be on the same chromosome, maybe even right next to each other. When they are close together, we say they are linked genes. A 2008 study demonstrated a link between human genes that influence hair color and skin pigmentation, including freckles. Moving on. Why are linked genes inherited together? To answer this question, we must revisit the behavior of chromosomes during the production of gametes discussed in Chapter 8. When a sperm or egg is produced by meiosis, only one of the two copies of each of your chromosomes ends up in the gamete. That copy comes from either your mother or your father. All of the alleles on the chromosome from one parent get passed on as a group to the child at fertilization. This process continues generation after generation. The linked alleles remain together unless during meiosis recombination occurs, exchanging one or more alleles with the other chromosome in the pair so that they now become linked with the alleles on that chromosome. When alleles are linked closely on the same chromosome, Mendel's second law doesn't hold true. It is surprising, and it was very fortunate for Mendel that of the 7 pea plant traits he chose for his studies, none were close together on the same chromosome. For this reason, they all behaved as if they weren't linked. And that brings us to our take-home message for section 9.17. Sometimes having one trait influences the presence of another trait. This is because the alleles for 2 genes are inherited and expressed almost as a package when the genes are located close together on the same chromosome.